Hello and welcome to Unit 1.4, Proteins. You're watching Freeology Revision. What are proteins? Proteins are one of the most abundant organic molecules in living systems. Amino acids are the monomers that make up proteins. All living things share a bank of only 20 amino acids. There are 20 different R, also known as variable, groups, and so there are 20 different amino acids. Since each R group is slightly different, each amino acid has different properties, and this in turn means that proteins can have a wide range of properties. Amino acids have a central carbon atom to which an amino group, a carboxyl group, a hydrogen atom, and a side chain, R group, are attached. You can now see an example of an amino acid on the screen. There is a central carbon atom. 4 things attached to this carbon atom. The central carbon atom is bonded to an amino group on one side, NH2. There is a central nitrogen atom bonded to 2 hydrogen atoms. On the other side, there is a carboxyl group, COOH. The central carbon atom is double bonded to 1 oxygen atom and also bonded to a hydroxyl group. Finally, there is a hydrogen atom, and an R, or variable, group. This is the basic structure of an amino acid. Now that we know the structure of amino acids, let's look at how they join together to form proteins. A condensation reaction between 2 amino acids forms a peptide bond. A molecule of water is released during this reaction. The reverse reaction, hydrolysis, happens during digestion. You can now see the formation of a peptide bond on screen. The hydroxyl group and one of the hydrogens from the amino group detach and form one molecule of water. The carbon from the carboxyl group and the nitrogen from the amino group form a bond. A peptide bond. Dipeptides are formed by the condensation of 2 amino acids. Polypeptides are formed by the condensation of many amino acids. The sequence of amino acids in a polypeptide chain is determined by the sequence of the genetic code on mRNA being translated in the ribosomes. A functional protein may contain 1 or more polypeptide chains. The shape of a protein is critical to its function. An enzyme, for example, can bind to a specific substrate at site known as the active site. If that active site is altered because of changes to the protein structure, the enzyme may be unable to bind to the substrate. To understand how the protein gets its final shape, you need to know the 4 levels of protein structure: primary, secondary, tertiary and quaternary. Primary structure. This is the first stage. It simply refers to a singular, polypeptide chain. There is a specific sequence of amino acids, determined by the specific sequence of DNA bases. Secondary structure. This is not specific. This is a result of protein folding. The secondary structure is held together by hydrogen bonds between the carboxyl groups and the amino groups in the polypeptide backbone. The backbone just refers to the polypeptide chain apart from the R groups, so all we mean here is that secondary structure does not involve R group atoms. The 2 most common secondary structures are the alpha helix and the beta pleated sheet, but it does not have to be either of these. An example of a protein with an alpha helix shape is keratin. One with a beta pleated sheet is silk. Next is the tertiary structure. This one is a little more complicated. This is further folding of the secondary structure into a 3-dimensional, globular shape. The tertiary structure is held together by bonds between the R groups of the amino acids in the protein, and so the shape depends on what the sequence of amino acids is. This means this type of structure is very specific because it is determined by the positions and sequences of R groups. There are three kinds of bonds involved. Hydrogen bonds, which are weak. They form between strongly polar groups. They can be broken by high temperature or by a change in pH. Ionic bonds. These are moderately strong. Ionic bonds occur between R groups with positive or negative charges. Ionized amine and carboxylic acid groups. They can be broken by pH changes. The strongest is disulfide bonds. Disulfide bonds form between molecules with sulfur in the R group. They are covalent sulfur-sulfur bonds. The bonds can be broken by reducing agents. Examples of proteins with tertiary structure include all enzymes. Finally, the fourth, quaternary structure. Quite simply, this means two or more polypeptides bonded together. In nature, some proteins are formed from several polypeptides, also known as subunits, and the interaction of these subunits forms the quaternary structure. Examples of this association of several polypeptides include collagen, a fibrous protein of 3 polypeptides; hemoglobin, a globular protein with 4 polypeptide chains; insulin, 2 polypeptide chains held together by disulfide bond. How do you test for proteins? If you need to find out if a substance, like a food sample, contains protein, you would use the biuret test. Step 1. The test solution needs to be alkaline, so you first need to add sodium or potassium hydroxide solution. Step 2. Next you add a few drops of copper sulfate solution. This is blue. Step 3. If protein is present, the solution turns purple. If there is no protein, it will remain blue. You may also need to be familiar with chromatography. You will do an experiment using chromatography paper during class. Amino acids have different R groups, which means they will have different solubilities in different solvents and can be separated by thin layer paper chromatography. A container is filled to a depth of about 2 cm with a suitable chromatography solvent. The container is sealed and left to allow the solvent to saturate the atmosphere. This is important because solvent must not evaporate from the chromatogram as it is running. A strip of chromatography paper has a pencil line drawn on it slightly more than 2 cm from the bottom, and small crosses are drawn on it to indicate where to add the amino acids. It is important not to touch the paper as there are amino acids on your fingers. A spot of amino acid is applied to a cross on the line using a capillary tube. The spot is dried. The strip of chromatography paper is placed in the container, making sure that the solvent doesn't splash above the line. The solvent is drawn up the paper by capillary action but does not evaporate from it due to the saturated air around it. The amino acid is carried by the solvent and each will travel at different speeds depending on their different solubility. When the solvent has nearly reached the top, the paper is removed and a line is drawn to show where the solvent has reached. This is the solvent front. The distance each spot has traveled from the center of the cross to the center of the spot is measured. The distance the solvent front has traveled from the cross is measured. This is used to calculate an RF value. Each amino acid will have a unique RF value, and as the RF values for all amino acids are known, they can be identified knowing the RF value. Let's recap. Proteins have a variety of functions within all living organisms. Amino acids are the monomers from which proteins are made. Amino acids have a central carbon atom to which an amino group, a carboxyl group, a hydrogen atom, and a side chain, R group, are attached. Dipeptides are formed by the condensation of 2 amino acids. Polypeptides are formed via condensation reactions between the amine group of one amino acid and the carboxyl group of another. A molecule of water is released during reaction. The bonds formed between amino acids are called peptide bonds. The reverse reaction happens during digestion. A functional protein may contain one or more polypeptides. There are 4 protein structures. Primary. This is a specific sequence of amino acids determined by a specific sequence of DNA bases. Secondary structure. This is a result of protein folding. The secondary structure is held together by hydrogen bonds between the carboxyl groups and the amino groups in the polypeptide backbone. The most common structures are an alpha helix and a beta pleated sheet. Tertiary structure. It is folding into a 3-dimensional, globular structure. The tertiary structure is held together by bonds between the R groups of the amino acids in the protein, and so depends on what the sequence of amino acids is, and so is very specific. There are 3 kinds of bonds involved: hydrogen bonds, which are weak, broken by heat and pH changes. Ionic bonds between R groups. These are broken by pH changes. Disulfide bonds. These are the strongest. They are broken by reducing agents. Quaternary structure. In nature, some proteins are formed from several polypeptides, also known as subunits, and the interaction of these subunits forms the quaternary structure. They contain the same bonds as previously mentioned. Testing for proteins. If you need to find out if a substance, like a food sample, contains protein, you would use the biuret test. The test solution needs to be alkaline, so you first need to add sodium or potassium hydroxide solution. Next you add a few drops of copper sulfate solution. If protein is present, the solution turns purple. If there is no protein, it will remain blue. Chromatography can be used to identify the different amino acids in a sample. Thank you for watching Freeology Revision.